Can you also see the screen?
Yes, sir, it's visible.
Okay, thanks. So, yeah, hi, I'm Arpit and I'm the TA for this course. And today we are doing the second live session. So I'll just get started. I hope other people will join. So here are some important links before we begin. So the first one is the uh, link for this session. The second one is the link for the YouTube channel where all these videos are being uploaded. And the third one is the Google Drive link where all the PPTs will be uploaded. So today we'll start with lecture 7 and 8. Before that, do you have any questions from the previous lectures? Any questions or any doubts? Okay, so one thing that people asked in the last session was how do you submit a scanned copy assignment? So I'm attaching a link to a YouTube video here. So you can check that. So that's a video from NPTEL where they show how to submit a subjective assignment. So just in case you guys need it. So anyways, we'll get started with lecture 7 and 8, numbers and scales in biology. So both the lectures are on the same topic. So I've just combined both of them so we can go through it together. So first, so I talked about what are the different types of scales that we generally use in biology. And we talked about the linear and the log scale. So we'll discuss both of them. So first is the linear scale. So a linear scale is basically like a simple number line that you have. So it's an additive scale. So what this means is, uh, so the major points that we have marked here on the number line are 0, 100, 200, 300, and so on. So to go from one major point to the next major point, you have to add something. In this case, you have to add 100. So if you add 100 from 0, you get 100. If you add 100 to that, you get 200 and so on. So this is the linear scale because all the major markings on the scale are basically additive, right? And then we might have markings in between these numbers. So for example, somewhere here, we'll have 50, somewhere here, we'll have 25 and so on. So how do we figure that out? So for example, if you have a linear scale and if you have, let's say, two markings, A and B, so the point here, which is in the middle, let's say it's Y. So the point Y in a linear scale is given as A plus B by 2. Right. So the middle point here would be 0 plus 100 by 2, which is 50. The middle point of these two would be 25 and so on. So this is how a linear scale works. I think most people are familiar with the linear scale. So we'll move on to the next one, which is a log scale. So now a log scale is what is known as a multiplicative scale. So what does this mean? So this means that to go from one major marking to the next, what we are doing in this case is we are multiplying something. In this case, we are multiplying 10. So 10 to the power minus 3 into 10 is 10 to the power minus 2, which is the next major point in the scale. And again, 10 to the power minus 2 into 10 gives us 10 to the power minus 1, or which is 1 by 10, which is the next major point. Then we have 10 to the 10 raised to power 0, which is 1, 10 raised to power 1, which is 10. Then we have 100 and so on. So the apparent benefit of using, or the most obvious benefit of using a log scale is when we want to refer to processes that are very diverse. So for example, like processes that might range from very small values to very large values, or measurements that might range from very small values to very large values. So for example, here you can see that in the linear scale, in this much area, we were able to cover only from 0 to 1,000. But in a similar kind of area, in a similar kind of space line, what we can cover here on the log scale is much larger. It's like orders of magnitude larger. So we are going from 10 raised by minus 3, which is 1 by 1000, right, to 10 raised by 8, which is like somewhere around a billion, right? Uh, so in the log scale, all these minor markings, as you might note, are not present equidistant to each other. So for example, in a linear scale, let's say we'll have here, we have 0 to 10. So 1 would be somewhere here, 5, and so on till 10. So all of these would be equidistant. But here in the log scale, that's not the case. So if you, so like visually, if you are seeing, so this is 10 raised to power minus 1. This point here is 2 into 10 raised to power minus 1. This point is 3 into 10 raised to power minus 1, and so on. So each of these markings are basically 1, 2, 3, and so on. So this will be, so this is 10 raised to power minus 1, which is 10. This is 2 into 10, which is 20. This will be 30, 40, 50, 60, so on until we have 100 here. Right. Now, if we are trying to read a general log scale, so let's say we have a log scale and we have 2 points marked A and B. So the midpoint here, let's say again we denote it by y. So for the log scale, the midpoint here y is given as A into B root over. So it's basically root over of A multiplied by B. So this is the midpoint. And now again, if you want to find the midpoint of this, which is y and b, so this part will be y into b root over. Is that clear? Does anyone have any doubts? Because these are like the most uh, fundamental things when we are talking about scales. Okay. So next we talk about what is the spatial scale of biology. So here we'll understand why we need a log scale when we are talking about biological processes or biological things. So many a times things go from very small to very large. So for example, uh, we have, let's say, an atom, which has a size of somewhere around zero point 0.1 nanometers. So a nanometer is 10 to the power minus 9 meters. So just write it down. 1 nanometer is equal to 10 to the power minus 9 meters. 1 micrometer is basically 10 to the power minus 6 meters. Then we have millimeter, which is 10 to the power minus 3. Centimeter, which is 10 to the power minus 2, and so on. Minus 1, yeah. 10 to the power minus 1. So 100 centimeters equals to a meter, yeah. So here we can see what are the different what are the sizes of different things that we often see around in biology or we talk about in biology. So starting from an atom, which is like 0.1 nanometers, we have like the DNA molecule, which has a width of around 2 nanometers. So the distance between each base pair is around 2 nanometers. Then we have proteins, which generally range from 1 to, let's say, 3 nanometers in size, but there are like some larger proteins also. Then we have viruses, which can range from 30 to 100 nanometers in size. Right. And then basically the next larger thing that we would have is basically bacteria. So for example, the size of a E. coli cell, which looks something like this, the dimensions of it would be like the width here would be 1 micrometer, whereas the length here would be 2 micrometers. So that's the size of a E. coli cell. So some of these sizes are important uh, for us to remember, or at least to remember a ballpark figure of these, because sometimes we might need to make some estimations, make some calculations like we saw later in the lectures, like we'll do estimation again. So basically, these sizes help us have a reference point from where we can start making calculations, right? Then we have mitochondria, which also has a size similar to E. coli. Then we have yeast, which is around 4 micrometers, human red blood cells, which range from which are around 7 to 8 micrometers, so approximately, let's say, 10 micrometers. Right, if you have trouble remembering 7 or 8, you can do this. Then we have cells, which range from 10 to 100 micrometers, like cells like animal cells and plant cells, not like bacteria and all. So plant cells generally are larger than animal cells, but animal cells also can have a very diverse range of sizes. Then we have things like paramecium and like other single-celled cells such as the human egg and the frog egg, which basically are much larger, which can, so for example, the frog egg can sometimes be around 1 millimeter. Then we have like common insects such as fruit flies and other uh, crickets and stuff, which basically are in the range of some millimeters to a centimeter, let's say. Then we have a chicken egg, which basically falls somewhere around 5 to 10 centimeters. And then we have different organs of the human body, which are much larger in the range of like centimeters and stuff. And then we have humans, which range from generally 1 to 2 meters. And even after humans, we have things like other larger animals, mammals, and all. And even things like elephants, blue whales, whale sharks, and all, which might sometimes be in the order of like tens of hundreds of meters, right? So this whole scale, this whole uh, range is what we see in biology, which is quite varied, going from like things like molecules and DNA and proteins to organisms. We see that there's a very large difference in the scale. And since biology studies all of these, it's very important for us to have a sense of idea of what the scale is and how different things, where different things fall on the scale. Right. So to put things into perspective, we can look at how big is a human cell. So we have here different kinds of human cells and their average volume. So for example, the smallest cell that we see is the sperm cell, which is around 30 micrometer cube in volume, whereas the largest cell that we have is the oocyte, which is around 4 million uh, micrometer cubes. Right. And the interesting thing is that both of these fuse to give rise to a zygote, which basically grows to become a human. And somewhere in the middle of this, we have this cell called the HeLa cell, which is basically a cancer cell line, which is used to study cancer. And basically, yeah, study cancer, how cancer cells grow, and how we can find drugs to cure cancer and things like that. So this abbreviation HeLa basically comes from a patient called Henrietta R-I-E-T-T-A Lacks, from whom these cervical cancer cells were taken. And there's a very interesting story which I think you should look up about how these cells were taken and uh, how they're used in labs and stuff. Okay. So here is a photo of a HeLa cell dividing. So this small tube that you see here is basically the last connecting link between these two cells, which will sever in sometime, and these cells will then be two separate individual cells. And the scale here is like, so this much is 10 micrometers. So all this is taken from this website called How Big is a Human Cell, which is like a part of the BioNumbers website. I urge you to go check it out and see and read even more interesting facts about cells and how uh, different kinds of numbers are present in different uh, basically areas of biology and things like that. Next, we talked about the temporal scale of things. So the previous thing was the spatial scale of things, or how uh, things are of different size and how uh, basically the size and shape of things. But the temporal scale of biology deals with how long event is, or what is the timing of a given event. So here again, we move from like large scale changes, such as changes at the level of ecosystem or population, which might happen over the course of a year. So a population size might decline over the years or increase over the years, or there might be changes in the ecosystem which might happen over centuries. So, for example, the community of a place changing, that might take more than a century to happen. Two other smaller things, such as organisms, organ systems, which basically deal with things at a temporal scale of days to minutes. So, organism has a kind of a circadian rhythm, which basically tells it when not tells it exactly, but determines in a way when you sleep, when you get up, and so on, which is generally aligned to the day, which is around 24 hours. Then we have organ systems and organs, which, for example, the digestive system, our stomach, kidneys, and all, which basically process food or generate waste. And they take around minutes or hours of time to basically do that. Then we have changes that happen at the level of tissue, which are processes that take place in tissues and cells, which might take like around seconds or milliseconds to happen. Then we have subcellular and molecular level things, for example, like DNA replication or steps of DNA replication, then cell division and so on, which might happen in like microseconds or nanoseconds. Like some of those steps might happen in microseconds or nanoseconds. Right. So the temporal scale of biology also ranges over a large order of magnitude, going from nanoseconds, which is like again seconds, and just by minus 6 seconds, and then we go to like a year or a century, which is like 100 years. Right. So we can also have a idea of like this temporal scale for a given organism. So, for example, if we take a human, we say that we have reflexes. So, for example, if you touch something very hot or cold, you immediately like retract your hand, right? Or when someone hits you in a specific place, for example, in the knees, there's a place where you specifically hit it slowly, your leg starts to move. So these things, these reflexes generally happen at the scale of, let's say, milliseconds or even microseconds at times, right? Then we have a heartbeat, which basically is around the scale of seconds. So normally a heart beats around 60 to 70 or 70 to 80 times per minute. It beats per minute. So basically it happens, one heartbeat basically happens close to 1 second. Then we have, as I talked about before, the circadian rhythm, which generally determines when we are sleeping, when we are waking up, when we feel tired, when we feel sleepy, and so on. This happens over the course of a day. So this is, let's say, around 1 day or 24 hours. Then humans also have a ovulation cycle, which basically happens over the course of 28 days or close to 1 month. Then we have a gestation cycle, which is basically when we have a baby in the stomach, in the womb. So gestation generally goes for 9 months in humans, which is again around a year. And we have a lifespan. So when the time between the birth and the death of a human, which is approximately 60 to 70 years, or ideally somewhere close to a century. So even in a human, the different processes or the different things that are happening at very different time scales, and we need to understand all this and basically remember all this. And because in science, the questions that we ask and the experiments that we try to develop and all will again depend on these uh, time scales. So if we want to look at the circadian rhythm of an animal, then we need to have an experimental design which basically takes into account that the circadian rhythm resets every day. Whereas if you want to test the ovulation cycle in some organism or the gestation period, then we need to be careful about how long we are carrying out our experiment for, because the time periods here are much larger and so on. Right. So next, so I talked about how we can read different kinds of scale. So for example, these two graphs are showing the same thing, which is the growth of bacteria in a medium. On the left-hand side, we have a regular graph where basically here time is in a linear scale, and here the number of cells is also in a linear scale. And we see that the growth of cells is non-linear because it increases slowly, but then after a point, it starts increasing quite fast. This happens because cell growth generally happens by in a multiplicative order. So one cell divides to become two, two becomes four, and so on. So the more number of cells that you have, the faster you generate even more cells, right? And now if the same thing we plot it in a semi-log graph, so now here the time here is again in a linear scale. So the week one, week two, week three, and so on. But the y-axis here, we have modified it to be in the log scale. So basically it goes from 10, 100,000 and so on. Here it's going from 0 to 25,000, 50,000 and so on. But here it's going 0, 10, 100,000 and so on. So here if we make a plot of this, we see that here we can fit a straight line because the log scale is also a multiplicative scale and the process of bacterial growth is also multiplicative, right? So we can plot a straight line here to show the growth, and basically the slope of this line, which is, um, let's say, which is m. So the slope here gives us what is called the doubling time, or the amount of time that the bacteria takes to double the number of cells that it has. So going from 2 to 4, how much time it takes, going from 4 to 8, how much time it takes, and so on. Right? So in the next lecture, what sir did was an estimation exercise, which we'll again do. So what is the number of cells in a human body? So let's say we are asked to approximate what is the number of cells in a human body. So one way to make this calculation, so for example, let's say number of cells is given as mass of the body divided by mass of an individual cell. Right. So let's say we have the average mass of a human body and we have the average mass of a human cell. We can divide both of that to get an approximate value of the number of cells in the human body. So the mass of the body, an approximate value or average value for the mass would be around 70 kgs. Right. But for the mass of a cell, we don't know. Right. So what can we do? So there's another equation that we can then start thinking about or try to look at. So the number of cells can also be given as the volume of the human body divided by the volume of a human cell. Right. Again, here we were dividing mass by mass. Now we are dividing volume by volume to get at the approximate number. Now we don't know the volume of a human body, right? So again, we are faced with a dilemma. Is there a way in which we can make some approximation, right? So again, for the volume of cell, how do we calculate the volume of cell? So if you have some approximations or if you make some simple assumptions, so for example, the first assumption that we make is, let's say, a cell, because we talked about scales and all those things before. what is the size of a cell, and so on. We talked about. So we know that the human RBC is around 8 micrometers. So let's say on an average, a human cell is around 10 micrometers. And let's say we assume that a human cell is a cube. Okay, so we are making a few simple assumptions. So the first thing is that the cell is a cube. The second thing is that uh, approximate cell size is around 10 micrometers. So now we have 10 micrometers, 10 micrometers, and 10 micrometers. So to calculate the volume of a cell, what we can do is basically 10 into 10 into 10 micrometers square cube, which is basically meter cube. Now, if we want to convert it into meters, so we want to make all the calculations in meters. So we know that 1 micrometer is equal to 10 power minus 6 meters. So 1 micrometer cube would be 10 raised power minus 18 meters, meter cube. Okay. So what we do is basically 10 raised power 3 into 10 raised power minus 18 meter cube, which is 10 raised power minus 15 meter cube. So now we have an idea of the volume of a cell. Now we can think about something called the density. So how do we relate like mass and volume? So that's the line of thinking, and we arrive at density. So we know that the density of anything is given as mass divided by volume. Right. So the volume of a cell is something that we approximated now. Now we want to figure out, so this part is done. Now we want to figure out what is the volume of a human body. So if we figure out what is the volume of human body, we can figure out what is the approximate number of cells. So we know that the mass of the human body is 70 kgs. And for the density, again, if we make another simple assumption, which is basically humans density is similar to water, because humans are 70% water. Right. So approximately the density that we have will be similar to water. So let's say we say that the density of a human is same as the density of water. So we know that the density of water is 1000 kg per meter cube. We know that the average mass of a human being is 70 kgs. So the volume of a human body or a human body. So what we can do is we can take this here and bring this here. So volume of a body is equal to 70 divided by 1000 meter cube. So basically 70 into 10 raised power minus 3 meter cube. So now we also have a value or approximate estimate for the volume of a human body. Now we can use this equation, the second one, to get at the number of cells. So the number of cells in a human body is equal to the volume of the body, which is 70 into 10 raised power minus 3 meter cube divided by volume of a cell, which is 10 raised power minus 15 meter cube. This is the volume of a cell. Then we have 70 into 10 raised power minus 3 into 10 raised power 15, which is 70 into 10 raised power 12, approximately 70 trillion. Now, if we go and look up what is the number of cells in the human body, it's somewhere around 37 trillion. That is the answer. So the value that we arrive at, which is 70 trillion, is almost in the same ballpark. So since we are making an estimation, we of course cannot arrive at the exact value, but we need to try and be as close to the actual value. So how can we make that happen? So to make that happen, we need to have reasonable assumptions. So to have reasonable assumptions, we need to have some basic idea of the size of different things, or we need to be able to come up with different ways of getting or estimating the size or the density or things like that for different things. Right. So this is an example of how we can do an estimation to get at what is the number of cells in the human body. Similarly, we can try and do a similar estimation for other animals, or we can try and see what is the size of a chromosome, what is the size of DNA, and like how many base pairs are present in the human DNA, and so on. If we know the size of the human chromosome and the size of the human DNA or the distance between each base pairs, and so on. Right. So that's where all these things that we talked about, the scales in biology, comes into play. So here we made the three simple assumptions. So the first thing is that the cell is a cube. That assumption we made so that it makes it easier for us to make all these calculations about the volume and so on. The otherwise, if we take any other shape, we can still do calculations, but it will be very complicated. Right? It will become slightly more complicated. And since we are doing an estimation, we don't need to be very accurate, so we can make some of these assumptions. The second assumption that we made is that the approximate size of a cell is around 10 micrometers. We made that assumption because we have an idea that the human red blood cell is somewhere around 8 micrometers, 7 to 8 micrometers. So it's reasonable that other cells are also of the same size. And the third assumption that we made was that the density of a human body is similar to the density of water. So that assumption we made because we know that humans are made up of mostly of water. Right. So if your assumptions are reasonable, the value that you arrive at will also be reasonable. And that is exactly the goal of this, the aim of this process. Does anyone have any questions or doubts? Okay. So when we are talking about numbers and scales and so on in biology, one very important uh, field of biology is where like numbers and scales are very important is something called allometry. So allometry basically is the study of how things in biology scale. So how basically, for example, in this case, what we are looking at is how the size of an organ increases with the body size of a organism, right? So how does the body size affect the organ size? So for example, this is for a human where we are basically making the plot in a log scale. So here we have log of body size in grams. Here we have log of organ size in grams. And we see that for the heart, which is the blue dots here, we see that there's a nice pattern. There's a straight, almost kind of a straight line which it follows. So that means that the equation describing the growth of, linking the growth of heart to the growth of body size will be something like log, let's say h is the size of heart, is given as alpha into log of b plus b, beta, right? Some constant. So this is like a straight line. So this is the equation of a straight line. So b here is body size, and h here is size of the heart, right? So we have a very simple linear equation that describes this phenomenon. So what it means is that the the other thing is, so the value of alpha is basically what determines how fast or how slow this organ is growing with respect to the body. For example, here, let's say approximately we see that this is somewhere close to the slope of this is somewhere close to 1, but we can say that the heart grows in sync with the body. Okay. Let's say this is approximately 1. So the heart grows in sync with the body. So as the body grows, the heart also grows proportionally. But if you look at the red dots here, which is the size of the brain as the size of the body increases. So this is from like a juvenile to an adult, what happens. So what we see is that the brain starts growing, but after some time it stops growing and the size of the brain becomes fixed. So there are a lot of limitations, a lot of physiological things that might be driving these things. So, for example, the size of the skull might not increase after a point. Maybe it's too uh, energy consuming to grow more brain. Maybe it's more difficult for neurons to divide and grow than it is for cells in the heart and so on. So basically many things, and basically these things are underlined by what part of the body needs to grow when, and how the body assigns different resources to different growing periods and so on. So basically allometry helps us deal and ask those kinds of questions and relate things such as growth of organ to growth of body. Another specific example of allometry is something called Kleber's law, where basically this guy called Kleber, he figured out that as the if we plot the log of a body mass along with the log of metabolic rate, which is basically the rate at which we consume energy in our body, right? So if we make that log plot, we see that most organisms, almost all organisms, fall in this straight line. Right. So going from even the smallest organisms such as a mouse, which is like a few centimeters in length, to an elephant, which is like few centimeters, like few grams or few milligrams, to going to elephants, which might be like tons, mm, which might have a body mass of a ton or more. We see that the all the organisms tend to follow this line. So that means there is some underlying relation between the body mass, and the metabolic rate of an individual or an organism. And this is also one of the areas of research where like, so this guy Kleber basically found out that this relationship exists, and that's why it's called Kleber's law, but no one knows why this happens. So there's an active area of research where people are trying to figure out why this is the case. Is there some uh, biological process linking body mass to metabolic rate, and like uh, how does like basically how this relationship comes about and so on. And why these kinds of things are important is because due to, especially due to things like climate change and all, people have found out across the world that sizes and shapes of different organisms are shrinking as the climate is getting warmer and warmer. So um, that's why these kinds of uh, laws are, and these kinds of allometric studies are more important, because we need to figure out like if this change in size and shape will affect the metabolic rate and the biology and the ecology of the species in some way. Right. So that's all for the first lecture. Does anyone have any questions or doubts? The first two lectures, actually. Okay, feel free to like unmute yourselves or uh, go to the chat box and post any question if you have any doubts. Okay. So the next lecture was basically a interview where basically uh, they talked about what is experimentation and what is the theory and how they are related and things like that. So Professor Sashi from NCBS was the one speaking as you, I presume, and basically they had this nice discussion. So what we'll do is I urge you guys to go and like look at the video and go through it so that you understand the whole thing much better. It's a nice interview. It's quite detailed, and like the ideas that they talk about are quite interesting. So what we'll do here is basically we'll have a short summary of the broad ideas. So I'm not going into much detail in this one, but we'll talk about the broad ideas that we talked about so that like it's easier for you to like distill and understand this information. So the first major point that was talked about is what is theory. So here Sashi sir gave an example of uh, basically what is theory. So he talked about how in ancient times and early times people tended to like do stargazing and all. So they noticed that there are certain stars which are present and which uh, across nights move in certain ways, right? From this, other people started figuring out interesting patterns of these movements. And basically, Kepler was the first one who put it into mathematical terms and figure out that certain celestial objects have periodicity and how they're related to each other in terms of when they appear in the sky and so on. And based on the mm, estimations or observations by Kepler, what Newton did was he proposed a theory, which is his law of gravitation, which tells us about how two bodies which are not in contact can exert a force on each other and how that force can lead to movement or basically, yeah, movement or orbiting of one body around the other and so on. So basically, due to Newton's law of gravitation, which was the theory that Newton gave, one could explain the periodicity and the relationships that Kepler formed, and one could see, explain, and predict, even predict the patterns that were seen in the sky. Right. So the main idea here is that uh, we have generally in science observations, and from those observations we try and elucidate some patterns, which basically gives us uh, some hint. And basically, after thinking about it, after like doing some experiments or so on, and trying to make some predictions and so on, what uh, some people, some scientists put forward is something called a theory, which basically is a mechanistic framework of why things are happening. So a theory is an organization of facts and data into a framework that can be used to explain the world or a process that is happening in the world. Right. The important thing is that theory is so basically we have observations, we have data, but theory is not exactly data. Theory is something that binds the data into a framework. So theory not only helps us to explain the world, but it also helps us to make certain predictions, both about what will happen in the future and also about systems that are similar to this. So, for example, Newton's law of gravitation tells us, like, helps us predict the future movement of planets. So now, like, we have things such as space bodies such as NASA and stuff, which basically regularly predict when like comets will come, when like we'll have. So recently we had a planetary alignment where basically all the nine planets were in the same plane or something. So things like this are things that can be predicted because of our understanding of Newton's law of gravitation, which is the theory. And even like in other systems, so even if we go move out of our solar system and move into other systems of orbiting bodies and so on, we can make predictions there because we know that gravity acts in the similar way even there, right? Talking specifically about biology, theory in biology, example of theory in biology is the theory of evolution by Charles Darwin. So what he did was he saw uh, on his trip on the Beagle, he saw a lot of different animals and plants and taxa. And what he did was he tried to understand why things are how they are. And based on his like understanding, he tried to develop a theory of evolution, which basically binds. things such as natural selection and heredity to give a more coherent idea of how many different species came about to be from a single species. So if you remember from the or from a single ancestral organism, right? So if you remember from the initial first lecture, I think first or second lecture, we had this beautiful diagram of a tree of life where we show basically how these different organisms are coming about from universal ancestor, which we call Luca. So basically, this theory of evolution by Darwin is how we basically explain the tree of life and how it came about to be. Right. Next major idea that was talked about in the video was how does theory and experiment feed into each other. So in this case, so I talked about a wonderful experiment carried out by Lurie and Delbruck. So what they did was basically, so at that time it was not known whether mutations occur randomly or mutations occur because of selection. Right. So if you have a certain stress in the environment, there's a mutation that occurs to deal with the stress, or do organisms have mutations, pre-existing mutations, which basically mm, lead to um, development of resistance to environmental stress. Right. So what they did was Lurie and Delbruck, they basically uh, made a theory. Right. They made a theory and they had basically two scenarios. So the first scenario is something called the acquired immunity hypothesis, where basically we are saying that after we uh, uh, introduce environmental stress or something, there is some mutation that is happening, which basically gives us, or that some change that is happening, which basically gives us some immunity to the environmental stress. And the second thing is the mutation hypothesis, where we say that random mutation hypothesis, where basically we say that mutations are randomly occurring in populations and sometimes these mutations can help deal with environmental conditions that are stressful. Right. So what they did was basically they took different cultures of bacteria as shown here, right? And they basically inoculated them for some time. So basically they grew the bacteria in the growth medium. And after that, what they did was they plated the bacteria on a plate which has some bacteriophage. So bacteriophage is basically a virus that kills the bacteria, right? And there are some bacteria which might have a mutation, which generally like, so it's also known that some bacteria have some mutation which helps them uh, fight off the phage and grow in plates where even phage is present. So what they did was they had these two different scenarios and they made some mathematical predictions based on the theory. So the first theory here is that the acquired immunity hypothesis states that basically once you do the plating from these cultures, after some time, after the bacteria comes in contact with the phage, it develops immunity. And therefore, you should see that if you do multiple platings of multiple colonies, you should see that on an average, X number of bacteria will become resistant. And the variance in the number of cells that are becoming resistant across the different plates would be very low. So the mean would be somewhere around the value X, and we'll have some deviation across it in each plate. Whereas in the second case, which is the random mutation hypothesis, what happens is some of these cultures will already have a mutation. So for example, this red bacteria here has a mutation which can help it to deal with the phage, right? And some of them will not. So for example, these are the normal bacteria which are not resistant. So once we do inoculation and we basically plate it in different phase, different plates having phases, what we see is we'll have very high variance because some of these inoculates will not be resistant at all. Some of them will have very high resistance. Some of them might have somewhere medium because of the number of bacteria that already have the pre-existing mutation. So what we should see here is that there should be a very high variance between the number of cells or the count of cells in each of the plates. So this is the framework or the theory that they had. So they had a hypothesis for this acquired immunity. They had a hypothesis for random mutation, and based on that, they were able to make predictions, mathematical predictions that if we do an experiment like this, we'll observe low variance. If we do an experiment like this and like this theory, so if we do an experiment, so the experiment in both the cases is the same. They're basically taking a bacteria, inoculating it for some time, and basically then plating it. So in the first case, we see that if we carry out this experiment and we see that we have low variance, then this theory is correct. And if we carry out this experiment and we see that there is a high variance, then this theory is correct. So what they figured out eventually is that the data has high variance. So this random mutation hypothesis is the one that is correct. So basically mutations occur randomly and there's no directed mutation that occurs. But what this example helps us understand is that they had some observations that basically some bacterial cells are resistant to phage, there are some bacterial cells which are not resistant to phages, and so on. They had some observations. Based on that, they developed like this theory, which is the acquired immunity hypothesis and the random mutation hypothesis. Based on these two theories, they were able to make predictions and design an experiment to test those predictions. So basically, it's like a closed loop. So where we have like some observations which help us generate a theory. Then based on the theory, we have some predictions, right? Then we do an experiment to test if our predictions are correct. If our predictions are correct, then our theory is okay, or it might sometimes our experiments might fail and we might figure out that the theory we have initially is wrong. So after the experiment, we have another set of observations which basically feed back into the theory and we can modify our theory and so. So this loop continues in science as we develop more and more specialized knowledge about different things. Right. Now, the next major idea in the lecture, in the discussion session was where do simulations fit in this? So a simulation is like a bridge between theory and experiment. So basically what we do in simulation is like modeling. So a model is in general a abstraction or an approximation of the real world. So what we are doing is we are taking some features of the real world that we think might be important in dealing with a problem or might be important in explaining a phenomenon, right? And we are basically taking only those few observations or those few features and trying to build a simple model of how things might be in real life. So simulations generally allow us to check theoretical ideas without having to perform costly experiments. So, um, for example, when we have very, what do you say, grand experimental ideas which might be very costly, which might require a lot of funding and so on. It might be a good idea to do carry out some simulations first and see if based on the theory that we have, based on the theoretical ideas that we have, do our experiments give us sufficient or good enough results, right? Now, if that happens, we can move on to doing the experiments. So a simulation can, as mentioned, can be a stepping stone for an experiment. Otherwise, sometimes what happens is it's very difficult to do certain kinds of experiments, right, in real life, because you cannot have, for example, experiments where you are trying to see cell growth in the absence of food. That does not make any sense, but like that's just an arbitrary example to show like there might be some experiments which you might want to do, but which might not be actually possible or which might be very difficult to do in real life, right? So in those situations, these simulations basically help us test out our theories or help us develop new theories based on, like, for example, we take a certain set of features or a certain set of ideas and we run a simulation and we can then see how things are moving and so on, and based on that how things are happening, how things are growing, and based on that we basically have modify our theory before we do an experiment and so on. So those were the three broad ideas from the discussion session. Does anyone have any questions or doubts? Okay, great. So next we move on to the next lecture where we talked about how to find a reliable information. So first we talked about and talked about how do we access information. So earlier people used to go to libraries and read books or written journals and magazines to get information. But nowadays we have the internet, which basically gives us a lot of power and makes a lot of information available at our fingertips, even from our devices such as a smartphone and so on. Right. But the issue with internet is that it has a lot of information. So there's a requirement for filtering information so that we only get what we want and what we need, and we don't get any useless information that is not relevant to us. So, for example, let's say we look at how chocolates are produced, right? So what we'll do here is I'll stop sharing this and I'll share screen. So I hope you guys can see this. So first what we can do is we can just look up about chocolates, right? So if we look at chocolates, we see that, okay, so there are these different kinds of chocolates, how much and does it cost and so on. Let's say we want to be more specific. So we want to look at more specific things such as chocolate production. Then we get these web pages which talk about how to make chocolate. Then we have this another one which talks about how is chocolate made and what it is made of. So let's open this and we see that, okay. So we see that this chocolate comes from this plant called cacao. Now, it's not a good idea to just look at one website and think that, okay, what they are seeing is correct. What we should be doing ideally is looking at many different websites and uh, getting at the general idea of what is happening. Right. Uh, so let's say we know, for example, that cacao is exactly how chocolate is made. Now we want to, let's say, talk about uh, cacao plant. How is it pollinated? So, sorry, pollination. So here we see that there are different uh, websites. So there are certain websites such as this chocolate journalist, cacao pollination, which uh, which seem like reputable, which seem like they are like good enough websites. And we have other websites such as the Scientific American or Science Friday, which might have some more scientific content. Now, if we are actually doing research and if we are trying to figure out scientific information, it's more important that instead of just doing a Google search, what we do is we look up information in something called Google Scholar. So Google Scholar, here we can go. So this scholar.google.com, here you can go and basically search for cacao plant pollination. And here what Google Scholar gives us is different kinds of different research papers. So where people have done experiments and published their work on cacao pollination and how it is pollinated and how we can increase pollination, increase the production, like cacao yield based on pollination and so on. So what we can do is we can click on one of these and open this. So if you click on the link here, it'll open a web page where basically you have an article which talks about uh, how cacao yield is limited by pollination and so on, if that is what you want to look at. So some of these articles will be free access or open access, so where you can basically read the whole article. So this is not. So the full text is not available. That's why it's not written here. But if you click here, so for some of these things, what Google Scholar has is like this feature where it shows like if there's a PDF for the paper available. So if you click here, it will open a PDF for the paper. So here we can read the whole paper, even if it is not available in the web page here. Great. In Google Scholar, you can have a much more specific search result. So for example, here, what we are doing is we are looking at articles that are published since any time. So we have articles from 1995 and we have articles from 2010, and even 1941, 1996, and 2017, and so on. But now let's say if you want a custom range, so if you want to just look at articles that were published in the last 2, 3 years. So let's say 2023 to 2025. And we search, it will give us only articles that are present in the recent years. So 2023, 2024, and so on. Again, we can sort it by date and we can look for different kinds of articles. So that's something we'll talk about slightly later. And yeah, so that's mostly it in Google Scholar. You can look for papers and you can open papers and read it. And we have another uh, oh, sorry. So the other thing is here you can click on related articles. So what it will show you is the first article. So this article, what all other articles are related to it. So maybe articles by the same authors, articles which are citing this articles which do a study that is similar to this article and so on. And here we have another option called cite. So if you click on cite, it basically gives you different formats of how you can cite this work. So in science, what is important is that whenever you are taking any information from other sources, so because you want it to be verifiable, you want it to be uh, you want like other people who want to know more about things that you're talking about. So you want to give them some reference of where you read it from, where you took it from, and so on. So for that, you have these references, which basically you should put when you are writing a report, writing a summary, writing a paper, writing whatever. Right. In general, it's a good practice to have references in bibliography. So when you press the cite option in Google Scholar, what it does is it gives you different formats of how you can cite the article in your work. So here it shows like who all are the authors. Then we have the title of the paper, which place or which journal it was published in, the volume of the journal, the page number, and so on. Sometimes it has the year and the date and so on. So these different formats will have slightly different information, but most of them will have the authors, the title of the paper, the journal where it was published, the volume of the page numbers, and so on for all these articles. So you can cite it so that anyone else who reads your written work can go and find this paper and read it if they are interested. So we have another repository called PubMed Central, where, for example, let's say we are looking for something called diabetes, where we are looking for diabetes. So we have like these different research articles that are coming up. So this PubMed Central is generally better for uh, things that are more in line with biology and molecular biology and so on. They have different kinds of databases apart from journal pages or research articles. So here you can select different database. So for example, you can look for books, you can look for nucleotide sequences, you can look for protein sequences, you can look for structure and taxonomy of organisms and so on in this portal. So PubMed Central. So this is also a very useful and important web page for researching and finding information when needed. So we go back to the slides. Right. So it is very important in science that we uh, are aware of something called plagiarism. So plagiarism is when you are basically copying stuff that other people have written without addressing who or where the original idea came from. And that's a very bad thing, and it's looked down upon in science. Um, so we should always try and avoid plagiarism. So the best things that you can do to avoid plagiarism is that don't copy full sentences from anywhere. What you should always do is read up a bunch of resources and write whatever you are writing in your own words, and then cite the resources that you have used to uh, basically write what you have written. Right. If needed, sometimes you might be quoting or copying sentences directly from other resources. If you do that, it's very important for you to quote the sentences that you have copied. So, for example, you put double quotes around here and you basically cite. Nowadays, because of development of AI, such as ChatGPT and so on, it has become very easy for students to basically plagiarize their mm, assignments and for researchers to write research articles or even other written material using ChatGPT and using AI. So it's important that in these cases, even when you are using, it's advisable to use ChatGPT as more of an editor than uh, a source where you get all your base written material, because in that way you are not understanding anything. So, for example, you go to ChatGPT, you look for a uh, answer to a assignment question and you basically copy the whole thing. You are not gaining anything out of it. So it's more important for you to read what is written there. Even if you are using ChatGPT, you can look for other reputable resources and read what is written, understand it, and try to write it in your own words. And it's also important to note that ChatGPT is not 100% right. And especially in the case of references, or if you are trying to look up scientific or factual information, it might give you incorrect information at times. So it's always important to double check your information before you are using it somewhere else. So we talked about how we can get citations from Google Scholar uh, and even from PubMed Central following a similar way. We can get citations. So I don't think I showed that. I'll just show that. Yeah. So here again, we can just click on this article. Let's say you want to read about diabetes. We go, we find this article. Then we have this button here, which says cite, and it will give us how to cite it. Right. So using both PubMed Central and um, Google Scholar, we can figure out how to, we can get basically citations for the works that we are using in our research. And this is an example of how you should cite things. So for example, here, when you're writing a sentence, which is in text, what you should do is you should write your sentence and then in brackets, write down where or which study says or which you got this idea from which study, right? And when you are having a reference or a bibliography at the end of your text or written material, you can cite the whole article like this. So this is the citation that we got from Google Scholar, or the full citation that we got from Google Scholar or PubMed Central. And this is basically how you should cite it in text. So there are these different citation or reference management software such as Zotero and Mendeley, which basically help you do this in a more structured way. So you can just go to a page and save the link to the web page on their uh, basically on their extensions or on their apps, and basically use that when you are writing in Word or when you are writing in Google Docs. So they have these integrations, which basically makes our life more easier. So that was it about how we can get reliable information from different resources. Does anyone have any questions or doubts? Okay. So we'll move on to the last lecture, which was on how to read a scientific article. So first of all, what is a scientific article? So a scientific article is basically a written up uh, format of what a researcher or someone figured out when they were doing an experiment, or basically a written up report of a theory or so on, right? So it's basically for us to publicize what we are doing and what we found out using the scientific method. So there are different kinds of articles. The first one is a research article where basically people do new experiments and write down the results of the experiments that they have. We have review articles, which basically um, are like a summary of the field, or basically a review article. To write a review article, what you do is you basically read a lot of different articles and then you try to summarize or distill all the information in those articles, so that you basically have so that for a given field or a given subdiscipline, you have like a general idea of where the field is, or you are basically in general in a review article proposing how in future people can progress by suggesting like some future direction of research or identifying like gaps in the research, right? And then we have something called opinion articles or commentaries, which are basically, for example, let's say you read a research article and you have an opinion about it. Let's say you agree with it, you don't agree with it, you find that it might be, it's a great article, you find that it's not a good article. So you can write an opinion article based on that. So for this course, we are focusing on peer-reviewed scientific articles. So what is peer review? What is the peer review process? So basically, peer review is when you are writing up and sending your work, uh, because what you're doing is mm, something very new. So the people who can understand and verify that are also very less, and they are exactly the people who are your peers or who are your colleagues who are working on similar things. So the peer review process basically ensures that uh, what you have written up, what you have done is basically the correct. So other people who are your peers basically review your work and say that whatever experiments you have done are good enough, whatever results or conclusions you are drawing from your study are good enough, so that you can get it published and share it with the whole wide world. Right. So the peer review process goes something like this. You basically conduct some experiment or write some theory or express an idea that you have. Then you submit that article to an appropriate journal. So for example, if I'm working on cancer cell research, I'll submit it to somewhere like the Journal of Cancer Research or something like that. I won't submit it to another journal, for example, Evolution or Ecology, right? Uh, whereas where if I'm working on something ecology related, I might submit it to a journal like Ecology Letters and so on. So once you submit an article to the appropriate journal, the editor then sends it out to editor basically goes through it and determines if it's a good fit for the journal, and then basically sends it out to different peers. to review it. Then the peers basically will go through what you have written, will try to replicate what you have done, and basically send you comments. You can address those comments and basically send the whole thing back to the editor, who will then decide whether it can be published or not, with or without any revisions that were suggested. So this is basically the peer review process, which helps us generate scientific articles and new knowledge and helps us propagate the new knowledge to other people. Note that not all articles are peer reviewed. So during the time of, especially during the time of COVID, uh, so engineering already had this thing, but for biology during the time of COVID, there was a lot of research happening on COVID-19, and when people were trying to develop uh, a vaccine for COVID-19, a lot of research was happening, and people were trying to basically uh, get this research across as quickly as possible. So what people came up with is something called bioarchive and medarchive, which are basically preprint servers. So here you can basically upload your work pre the peer review process. So the articles that are published in bioarchive are not peer reviewed, but basically they are published so that other people can quickly look at your results and see what is happening and maybe give you comments and feedback on your work before you submit it for peer review. And some people even don't go for peer review and just submit their work in preprint servers where basically they get comments from other people and maybe incorporate that. So how does a research article look? So this is an example of how a research article looks. So we have uh, here, which is the title of the article. Then we have who are the authors of the article. Then we have this thing, something called the DOI, which is a digital object identifier, which basically is a link. Sorry. Which basically is a link pointing to the article. It is a fixed link which does not change. Then we have, sorry, on top of here, we have the name of the journal where the article is present. Here, in this case, it's PLOS ONE. Then we have other options such as download the article, print it. Uh, we can look at how many people have cited this article, how many people have viewed and shared this article, and so on. When it was published, sorry, then we see that it's an open access article, so that means it's available to everyone. We see that it is peer reviewed, so that means it has gone through the peer review process, and it is a research article, not a review article. There's other information such as, then there's the article, which will come to. So this is how the article looks if you download it. So again, we have the title, the authors. So this 1 and 2 basically tells us where the authors are from. So you can figure out where the research is happening. So this is happening in Center for Ecological Sciences, Indian Institute of Science. And also like some authors are there from Department of Mechanical Engineering, again from Indian Institute of Science. So the first thing that you generally see in an article is the abstract. So the abstract gives you, so it might not always be structured in the same way as this. It might just be a bunch of paragraphs. It might be a single paragraph. But what an abstract gives you is basically some background of the article, some background that is needed to understand the work, then what work was done, and in brief, what results the researchers found out. The abstract is like a shortened version of the whole paper. So this, when you look at the abstract of a paper, you can immediately know if you are interested in the article, if you want to read the whole article, or if you feel like, okay, this is something that I know, or this is something that I don't need to know, so I don't need to read it. Right. So it can help you screen articles. So this, so for the course, we have a website called I Think Biology. So I've linked to this here in the bottom here. So basically, we have this section where we have in the website something called research highlights. where basically they have highlighted research from different mm, research and different studies, and given like a small summary of the whole thing, of summarize the whole content of the research paper. Then you can also download an annotated paper from here by clicking on this button. If you download that paper, so earlier we saw a PDF which was not annotated, but this is a PDF which was downloaded from I think Biology website, and it is annotated. So what it does is basically for each of these words, each of these difficult words or processes or things, it has simple definitions and highlights core ideas of the paper so that it's easier for you to read the paper. I would suggest like going through this, I think, biology website and looking at the research highlights and going through all the papers and seeing like if you can understand these papers on your own or if you need any help. And if you need any help, we are always here. And the research, the annotated paper can help you a lot in understanding these different simple terms that are used in the paper or understanding difficult jargon that might be used in the paper and summarizing the broad ideas of the paper. Right. So this is how a research paper is generally organized. So first, we talked about what we have is the abstract, which is like a short summary of the work. Then we have the introduction, which provides us the background of what are they looking at, why it is important, where does it fit in the larger scheme of things, and so on. Then we have methods or material or methods, which uh, basically makes something replicable. So in the method section, researchers generally give detailed instructions on how to replicate their work and how to generate the figures or the results that they have generated. So, so that other people who are reviewing the work can basically replicate it, or you, if you want to replicate what they did, you can go and replicate the same thing and find out if the result that they have figured out is correct or not, right? Or if there is some other method that you might that other improvement or the thing that you can do to the methods. Then we have a result and discussion section where we basically have the results of whatever experiment was carried out, and then a discussion of how those results fit into the background or fit into a larger scheme of things. So how do we read an article? So scientific journal articles are very dense. They require a lot of patience and can be read in a lot of different ways. Ma'am talked about one way of reading the paper, which I'll also reiterate. But you must note that it can be read in a lot of different ways. So if you want, you can try out different things and see what works for you. And when you are initially starting to read papers, I would suggest it's very important that you read and reread papers so that you understand and get the idea and get the hang of how to read papers. Right. And it's also suggested that you can go to this link. I think biology research highlights, go there and look for different research papers and the research highlight and download the annotated papers, because these might be a good starting point for you to start reading research papers, right? Because these are annotated and you have all the information that you need to understand it easily available to you. Right. So the process that ma'am describes, I'll describe that to read research papers. So the first thing is to read the title and the abstract of the paper. So sometimes it might be better to do this as a group and to of course always when you're reading the title and the abstract, try to clarify your doubts, try to look up for words or expressions that you might find it's difficult or uh, you don't know. For example, in this article as we so if you are reading this article, this abstract for the first time, you might come across many different words such as sclerotization, ovipositors, uh, trophic ecology, mechanoreceptors, which you might not know about. So it's very important for you to go look for these words, what they mean, and so on. And in general, it's a good idea to have a glossary of things. So you basically have a kind of a small dictionary for yourself where you are basically writing down things or concepts which you are encountering in research papers, and keep a habit of doing that so that will eventually help it make it easier for you to read research papers and help you understand research papers much more easily and understand concepts much more easily. The next thing after reading the title and abstract is to skip to materials and methods. So once you read the abstract, you have a basic idea of what is the background, why they are doing the research, and a brief overview of what they did and what they found out. Now, when you go to the materials and methods, you can see in detail what they are doing. It's not important for you to understand all that detail, but have an overview of what exactly they are doing. So, for example, uh, you might not. So, for example, if we read this paper, okay, so we don't have that. So, for example, what do I say? So, for example, they are saying that a specific kind of test was used to figure out the amount of. So, for example, you are reading a paper on diabetes, and you see that a specific kind of test was done to figure out the blood glucose level of different participants of the study, and so on. So, you don't need to know exactly what that test is and how it is done, but you need to know that the test was done to figure out the blood glucose levels and what was done with the blood glucose levels once it was figured out. Right. So ignore some of the things that you are not understanding, but try to have an idea of overall what they are doing. So ensure that you understand the overall goal of each method and you need not focus on the detail. And as I said before, start making a glossary of terms that you are not understanding using the internet. Then you move on to figures and figure captions. So figures are generally one of the most detailed parts of the research paper, where basically they're uh, so in general, when people are writing research articles, they write it in a way so that the figures are standalone. So that if someone comes and reads the research paper, they only need to go through all the figures to understand what is exactly happening. Right. And they don't need to uh, like they need to read the research paper, but even reading the figures gives you a good idea of what research was carried out and what they found out. So use reading the figures and figure captions, you can see uh, what are the testing and what method they are using, what are they presenting, how they are presenting the results, what are the results that they found, and what is the conclusion that they found from the results. What are the inferences that they are drawing from the results. Then, after doing all this, after you read the abstract, you read the materials and methods, you read the figures, you see the figures properly and read the figure captions, go back and read the whole paper. So now you have the main ideas. So though the paper, like some papers may address several sub questions, when you are reading, try to summarize the broad overarching major questions or problems that the authors are addressing and trying to answer. It is also good to try and summarize your reading to a peer. So, for example, you read a paper, then you try to explain it to one of your friends, or two or three of you read the same paper and try to explain it to each other, and so on. So, what happens here is when you have a discussion about a paper, when you try to explain a paper, other people might ask you questions which might make you think about things that you have not thought before, which might make it clear to you that there might be certain things that you might not have understood in the first time when you were reading the paper. So when you try to explain it to them, you see that, okay, so this is the part that I was not clear about. Maybe if I read it once more, I can explain it better. Maybe I can understand the paper better after that, and so on. Right. So It's also suggested that you make a summary or you basically present a paper that you are reading. So this is also one of the things that happens in general in a lot of labs where they have different journal clubs where you are basically some people basically read a few papers or read some paper and then explain it in detail to other people in the lab. So this is basically a suggested format for, and then they have a discussion on that. So this is basically a suggested format for the summary of the paper or for making a presentation out of a paper. So when you're looking at the introduction, you should use the background information to give some context. Why what was this? Why was this an important discovery? What was discovered? What happened? Then what is the history behind it? So like how people started asking this question, why people were interested in it, and so on, and introduce one overarching or major question or problem that the authors are addressing in the research article. Then you have like subheadings, and you're basically using figures as a guideline to discuss what question was addressed, which experiments were done to address the questions, and so on. And then when you move on to the conclusion and discussion, try to critically assess the paper by seeing if the major questions that were posed by the authors in the introduction were addressed in the results and discussions. So, do their results support their conclusion? Do their results tie up with the introduction and so on? Did they do all the necessary control experiments? How was the experimental design? Was it good? Do you think you can do better? Do you think they did a wonderful job and so on? And why they did a wonderful job and so on? So these are the things that you should think about when you are um, writing a summary of a paper or you're making a presentation. Why is this result important? How does it help uh, and add to a larger body of work? What else could be done? What else did the authors miss out on? What did the authors figure out, which was very interesting, and so on? And then trying to answer questions like what could be done next? So this study has been done, then what could be done next? So for example, the windmill study that we talked about in the first live session. So the windmill study is figured out by Maria Thakur and all, et al. Figured out that basically windmills lead to this increase in the um, abundance of lizards. Right. So next, you can try and think about, okay, so what else can we look at? What next can we do? Right. So they just looked at the lizards, but they did not look at the birds in detail. So maybe you can look at the birds. Why are the bird populations affected? Is it because they are basically getting, what do you say, hit by the windmills, or is it because they are making nest on the windmills which are falling down and the eggs are like not able to hatch, the eggs are getting destroyed and so on. So why all these things are happening? So what next could be done and so on? I think that you can think about. So that's it for the lectures. So does anyone have any questions or doubts till now? Okay. If no one has any questions or doubts, we'll move on to practice assignment 2. So we have 5 questions to discuss today. The first thing is Theomargarita magnifica is about 1 centimeter long and is one of the largest bacteria discovered in the world. It is considered 5,000 times larger than an average microbe. From this information, can you calculate what is the size of an average microbe? Can anyone answer this? Or can anyone tell me how we can solve this? Don't worry, you're not getting judged or getting any marks or anything based on this. So you can just try and answer. Just write anything in the chat or unmute yourself. Okay, so do you guys have any idea of how we can try and deal with it? Don't worry, just like and answer. Okay, so I'll solve this first one. Maybe you can try and answer the next one. Right. So it is mentioned that the size of this bacteria is about 1 centimeter. 1 centimeter. And it is considered 5,000 times larger than an average microbe. So the size of an average microbe should be 1 centimeter, the size of this divided by 5,000, because it's 5,000 times the size of an average microbe, right? So when we do this, so these are basically micrometers, these are millimeters. So when we do this, since we have two options, we have options in millimeters and micrometers. Yeah, what we can do is we can basically solve it in both micrometers and millimeters. So it's 10 millimeter, 1 centimeter is 10 millimeter divided by 5,000. And 1 centimeter is also equal to 10,000 micrometers divided by 5,000. So what we have here, if we do this division, is we get 2 into 10 power minus 3 millimeters, which is equal to 2 micrometers. So this is nothing but 0.002 millimeters and 2 micrometers. So we see that the option here, option A, 0.002 millimeters, and option D, 2 micrometers, are both correct. The answer here will be A and D. Okay. So next, in the discussion with Dr. Sashir Pudupalli, a theory is A, collection of data and facts, B, organizing data and facts, C, a conjecture without any factual basis, D, an organization of data and facts into a framework that can be used to explain the world. So what do you think is the correct answer? A, B, C, D? Can you guys just type it into the chat box or unmute yourself and speak? Okay. So the answer here would be option D, which is an organization of data and facts into a framework that can be used to explain the world. So that is what a theory is. We covered this when we were talking about this lecture a while back. The next question is, what is a critical step in assessing the credibility of scientific information found online? Option A, accepting information that aligns with your hypothesis. Option B, checking for peer-reviewed sources and publications. Option C, preferring information from newly established websites. And option D, information backed by reputable scientific journals or institutions. So what do you think is the correct answer here? We are talking about how we can assess the credibility of scientific information. Okay, so here accepting information that aligns with a hypothesis is the wrong answer, because we need to find information that is true and credible, not something that only aligns with your thinking. The second thing is checking for peer-reviewed source and publication. That is correct. That's a critical step because we want to look for information that is peer reviewed and other people have verified it. Preferring information from newly established website, this is not a critical step. Newly established websites might be right, might be wrong. There's a chance. And option D, information backed by reputable scientific journals or institutions. So this is correct. So the answer here will be B and D. The next question is, what is the importance of a literature review in a scientific article? A, providing an overview of existing research on the topic. B, to identify gaps in current knowledge or areas for further research. C, sharing the author's opinions about the research. And D, speculating about potential future research. We talked about this. What do you think is the answer for this? Okay. So a review article or a literature review basically provides an overview of what existing research is there on a given topic. It also helps us identify gaps in current knowledge and find out areas which can be where further research can be done. It's not opinion piece. It does not share opinion about the author's opinion about certain research, and it does not speculate about what potential future research is going to be carried out. It just gives us a direction of what potential research, what future research can be carried out. Okay, it's not speculating about what's going to happen. It's about what you can do, what people can try and look at. The fifth question is an abstract in a scientific paper is A, summary of the background, methods, findings, and implications of the scientific study. B, a summary of the literature review and research questions of the study. C, an outline of the methods used in research. And D, a summary of the author's personal opinions. A very simple one. Does anyone have an answer? Okay. So the answer here will be A. So an abstract is basically a summary of all the different sections of a paper, including the background, methods, findings, and the discussion or implications of the scientific study. It is not a summary of just the literature review and research question. It is not an outline of just the methods, and it is not anything, it does not concern anything to do with the author's personal opinions. Okay. So that's it for today. Does anyone have any questions? In today's session, we talked about numbers and scales in biology. So the spatial scale of biology, linear and log scale, temporal scale of biology, allometry. Then we talked about experimentation and theory. What is theory? How theory, experiments, and simulations fit in the broad scope of science. We talked about how we can find reliable information. We looked at examples including Google Scholar and PubMed Central. We'll talk about plagiarism, how to cite articles. Then we talked about how we can read scientific articles, what are different kinds of scientific articles, what is the peer review process, uh, what are preprints, how are articles structured. Then we talked about the organization of a research paper, how to read a research paper, and yeah, then we had some practice assignment and questions to solve. Does anyone have any questions based on whatever was taught or discussed today? Please feel free to ask your questions, unmute yourselves, or type it in the chat box if you have any questions. Okay, if no one has any questions, we'll stop here today and we'll meet again next time, next week, same time. Thank you for attending. I hope you'll attend the next class also. Okay, I'll see you next time. Mm. Bye.